Okay, we are live.
Thanks, Rishi.
No problem, buddy.
Rishi, can you get Michael to just announce in that room that life science is in here?
Yeah.
Cool. And then we'll go ahead and start. What's up? YouTube minutes? Yeah, you can start your own channel. Hey, it's back for another YouTube video.
I know.
What's up? I have a question. What is life?
It's not the purpose of this presentation. Life science. They should make it. No, they shouldn't. Philosophy. Okay, so we're gonna go ahead and get started. This is life science events. We'll go over like anatomy, disease detectives, genetics, and just whatever else we have time for. And at any point in the presentation, y'all can ask us any questions. So our presenters are.
I'm Vaishnavi. I was last year's anatomy and physiology supervisor, and I'm the lab events coordinator for ATXO.
And I'm Ashish. I'm the president of ATXO. So basically, with an overview of the life sciences, that in general they tend to have a low number of cheat sheets allowed. So you kind of have to be efficient in your space, and they kind of expect a lot of that information to be in your brain. For taxonomy events, a lot of the times they let you bring like a binder, or a lot of the physics events like a binder. But for life science events, you have to be a little bit more efficient with your studying. And yeah, so these are highly memorization based and context clues based. And the overall test quality actually tends to be better because for the most part, there are a lot more people who have expertise in the life sciences who are able to write good tests for these at the invitational level. So Vaishnavi will start us off with, First, I wanted to ask, like, what are y'all's current strategies to approach like studying for a life science events? And this is geared to both like coaches if you have observations from your students or students as well. Is there anything y'all do differently for life science events compared to like other events you're studying for now? So basically what I used to do was make sure that I had like a running Google document of a bunch of different terms and strategies. I would create like a Quizlet to help me memorize the things that needed to be memorized. And then I would also try to continue to pare down that knowledge, and so that I could continue reading back over and eliminate stuff that was now like obvious and given to me. So therefore, I could eliminate the knowledge that I needed to put on the notes to like very little. What about you? Do you have any strategies in particular besides what I just talked about?
Just being very particular what you put on your notes sheets. I think because you have such a low amount, you want to make sure that every single thing on your notes sheet can be used during the test. So, making sure you're not copying the entire wiki onto your notes sheet in small font, right? And just making sure you know exactly what you're putting on there.
Cool. So, Vaishnavi will start us off with anatomy and physiology.
So this is okay.
Sorry.
Is this right?
Yeah.
Okay. So this year's anatomy and physiology topics are the integumentary system, which is skin, skeletal, and muscular. So something, even though they give us these three categories, we have to make sure that we're not just sticking to them. For example, what I would recommend is co-studying systems. So, for example, if you were doing the skeletal system, what system goes along with the skeletal system? The muscular system. So you wouldn't just be studying skeletal conditions or muscular conditions, you would also have to study skeletal-muscular conditions. So that's just how the whole body works, right? Everything is intertwined with each other, so that's also how we want to study for this test. Also with the integumentary system, that is skin. But what's a system that relates to the integumentary system? Or something that might act with the same conditions and things like that? Not exactly. So something that has a similar function is the excretory system. So something similar in that way. So they could ask you things that are similar in condition or similar in function. So if you're studying the integumentary system, I would also recommend brushing up a little bit on the functions of the excretory system. So that's something that they don't really mention in the guide, but I would definitely look out for that. Let's see if there's a question on that. Also, the main skills with anatomy and physiology are we like to observe, make inferences, and make predictions. Anything that involves memorization can be put on a sheet, right? The things that we want to gain the most points on are being able to make sure that we know how those terms relate to each other and how we can put it on the test and use it for different questions. So I'm going to slide on note taking because I feel like this is the most crucial part of this event. So as you know, do you guys know how many pages you're allowed for anatomy? 1 page. Back and forth, that's it. That's crazy. So I made some recommendations on what I would take notes on and what I wouldn't take notes on. So what I would be taking notes on is anything that you feel like you need to memorize. So these are major terms related to the system, major conditions, symptoms, and something that people always miss out on are tests. So that's like MRIs, biopsies, especially for this year's, if you were talking about the integumentary system, you would mainly want to study biopsies. So things like that. And diagrams, big red flag diagrams. Your test will always be 25 to 50% diagrams. That's where the points are. So you want to make sure that these diagrams are on your note sheet. So I have a few slides on diagrams later. But what not to be taking notes on, I would definitely not copy the entire wiki because the entire wiki is not going to be helpful and it has a lot of fluff information that you don't need. I would not put or write stuff about inferences that you can make from a patient's condition. So when you write all these terms down on your notes sheet, the definitions itself will tell you or will help you make a prediction of what exactly a patient has. I definitely wouldn't put entire case studies or relationships between terms and conditions. So those are the things that you need to really hone in on and focus on when you study. So I would not exactly focus on memorizing all your terms, but I would focus on where the relationship between these terms. And so that's something that should definitely be practiced over and over again. So I want to talk about the type of questions on this exam. I know you guys have probably looked at a couple practice exams, but I have a couple questions that are related to the systems that we're doing this year. So we always have matching questions. So those are your easy point questions, right? Because those terms you have on your notes sheet, or you should have on your notes sheet. So those are your basic terms and everything. Diagrams, really important. Make sure you have any diagram related to your system on your notes sheet. And they will ask you every single part of that diagram. So you want to find a diagram that's very detailed. Something that I've noticed with anatomy and physiology students and the test is that the diagrams that come in the wiki are not as detailed as they should be. So you need to actually go to Google and find really detailed diagrams that show you every single part of the system. Because the ones on the wiki also don't match what's on the test. So you just want to make sure you're familiar with all types of diagrams as well. And then we have visuals. So for this year, this is going to be super important. What I'm guessing is that they're going to give you a visual of a condition, and you're going to have to tell them what it is and the symptoms, right? So we don't only want to know the name of the condition, we want to know what it looks like. So that's something that you should be studying. I don't think you need to put on your notes sheet, just make sure that you know what exactly it looks like. So, and that is about that for this slide. And then for the next slide, so now you have short answer-like questions, but they're in multiple choice format. So for this question up here, it seems like a lot of words. But basically all it's asking you is to know the definition of the 4 different types of skin, or the 4 different layers of skin. So these type of questions can be tackled really easily. All you have to, what I would recommend for these kind of questions is bringing a highlighter to the event. So especially with students, when they get to the test, they see a lot of info and they freak out and they don't actually read everything. So to make things a little bit easier, I would recommend bringing a highlighter and actually highlighting the information. So from afar, this question looks like a lot, but at the end of the day, it's just which skin layer is it. So I would recommend bringing a highlighter to actually dissect the question up. And then you have easy questions like, which of the following is not a layer associated with skin? So you should have those on your diagrams or somewhere on your note sheet. And then, things like listing the hair growth cycle, listing different layers, stuff like that, you'll most likely see on the exam. Okay, diagrams are super, 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 super important. I can't emphasize enough how important they are. I have put like maybe 8 diagrams up here that should probably be on your notes sheet. So we have the muscle cell diagram. You will most likely 95% get a question on that, I'm pretty sure. You have the muscle cell diagram. For skeletal system, we don't really think of diagrams. We think we only need to know the bone structure, but there are levels to bone structure that you need to know. And then for the skin cell, that will most definitely be on the test. Different layers, what each layer does, what the burns cause. So definitely need to know those layers of the skin. And then this is also another level of the muscle cell diagram that's very important. Something that kids don't usually study is this diagram especially. It talks about the Z disc, the M line, and thin filament, elastic filament. This is usually a question that comes on the test that kids are not prepared for. So I would definitely make sure that you have that diagram on the test. And then usually for muscular and skeletal, they'll give you a diagram of maybe a human body and say, okay, which muscle is this, which bone is this, and so you want that generic diagram on your notes sheet as well. And then also another thing kids miss is the nail diagram. Nails are also really important. So that will fall under the integumentary system. And then you have hair follicle diagrams. And also, do you guys have any ideas on what kind of pictures they might show on the exam in relation to diseases or conditions? So they might just give you a picture of something and ask you, what is it? Do you guys have any ideas for this specific category, what they would be? So something really important when it comes to the integumentary system is cancer. They'll give you different pictures or they might give you different pictures of cancers and ask you to identify what they are. Because when it comes to the integumentary system, those are the only visual aspects of cancer. So they might give you 3 different moles or 3 different colors of skin, and they might ask you what cancer is this or what does this look like. So that's really important when it comes to the skin system. So overall, I feel like skin, it feels very simple, but it's the most complicated part of this exam, for sure. Okay, so now we have the biggest question on your test, the case studies. So the case studies are huge, definitely from this length to maybe half a page. That's how big case studies are on the exam. So when we look at case study questions, the first thing we see is, or the first thing students see is probably a bunch of blah. They see so many words, they see all these questions, some symptoms about patients, they don't really know what's going on. So what would be your approach to approaching this question based on what we talked about? Like, what would be the first thing you would do as soon as you saw it? Highlight, like literally highlight any portion that you don't know, highlight any portion that's relevant to the question. Because I'm telling you, on the anatomy and physiology exams, anything you write that's even remotely relevant to your question will at least get you 1/4 of a point, right? Because the exam is so complicated and the answers are so complicated that you've got to put something down that's related to it, and you will at least get 1/4 of a point. So making sure that you're even understanding and reading this question is really, really important when it comes to this exam. So you guys are right, we highlight everything. And so that's just an example of what exactly you would highlight. And then these are some examples of the questions that could come from the case study. So everything here is something that you would probably not have on your notes sheet except for the biopsy in the third question. So in the first question, that's more of a prediction, right? So on your notes sheet, you would have all the terms and you would have all the diseases on there, but you would have to know your symptoms and what you would predict for that specific disease. And then for the second question, that's also kind of a prediction inference question. And then for the third question, that's where your notes sheet should really help you out because it talks about biopsies, and biopsies should be on your notes sheet. So then again, just reiterating, what we need on there, what we don't need on there is really important because we only have one page front and back. Okay, so one thing I really wanted to talk about was the grading for this exam. I feel like this is not really stressed on, but this exam is all partial points. Anything you do on this exam, even if you write 1 or 2 words, partial points is viable. So this exam, I think, is not a lot of multiple choice. Most of the time it's a lot of free response. Your easy points will come from the diagrams, right? Because you have the diagrams on your note sheet, you know what they are, just put them on the paper. Your hard points are going to be the case studies. So those are going to be where you want to try to get partial points. So even if you don't know what the condition is, what the disease is, write something about it. Like, what do you think it is? Based on the question, maybe what part of the system is it? Just write anything you have down because you might get partial points for it based on the grading. So that's something I just really want to emphasize. Don't leave it blank. Like, I know everybody says that for every test, but like, especially for this exam, you do not want to leave it blank. I'm telling you, you will get points if your answer is related to the answer on the key. And then the questions that get you the most small points are your diagrams, your multiple choice, things like that. Your multiple choice will generally be very straightforward, nothing too crazy. But where the hard points are the case studies. So I would make sure that you guys know that. And just a final thing is prepping for this exam. So definitely starting early and making sure that you're testing often. So Quizlet, great resource for this exam. Quizlets that other people have made, also great resources for this exam. You never know what you missed, right? So when I was doing this event a long time ago, I used to make my own Quizlet, and then I used to search up other people's Quizlets to see if I was missing any information. So that's something that I would recommend doing. And then understanding the material and not memorizing it, because you have a notes sheet for that. There's no need to stress over memorizing everything that could be on the exam. And then actually, I read something in the manual about there being specific handouts about the skeletal system and the muscular system that you need to know to make sure that you're not like over studying. So that's kind of a screenshot I got from there. They should have a 2019 or 2018 version, but I'm not exactly sure. But yeah, they have handouts for that. So I would definitely look at those handouts and then probably use the diagrams from that for your note sheet and not from the wiki. And then your resources, you've got like your textbooks, WebMD, most of the Science Olympiad websites and everything. And this was just a screenshot I took. You guys probably all have this manual, right? Everyone's got it. Just really important things to know. 1 sheet of paper, double-sided, non-programmable calculator. I do not think there will be a lot of formulas on this exam specifically because we always see that on cardiovascular, but I would have a non-programmable calculator just in case. I have 50 minutes and it's 2 people. And that is about it. Does anybody have any questions for anatomy? I know it's a really stressful exam. It's really stressful for the kids too, but definitely if you take your time, make sure you're splitting the exam between the 2 team members and working on it. I think that's a really good thing.
So we have 15 members who
Right.
are in 6th grade, so
Right.
it's first time for them.
Mm-hmm.
So what do you recommend for 6th graders?
For 6th graders, I would definitely recommend having, I think where they get the most points is on their diagrams.
Okay.
So I would definitely recommend for 6th graders to make sure they have all those diagrams down on the note sheet. And I would recommend them studying the terms. So we have a lot of terminology on the exam, and I think for Division B that's especially important. version of it. For Division C, we talk about more inferences, more case studies, and yeah, so definitely that's where you want to focus on. Yeah, for sure. Also, you know, if your students have been doing the anatomy and physiology exam like 1, 2, 3 years, that's really good because they end up learning how all the systems work, right? Because every year it's different systems. So, you know, getting them interested and then repeating it over and over again is actually a really good idea.
Okay.
So, yeah, thank you.
Thanks, Vaishnavi.
Thank you.
So I just want to really actually drive home how cool I think these life science events are, especially anatomy and disease detectives. Right now I'm in an EMT training course, which is just like a semester-long thing to become certified as an EMT, and they always ask us like, you know, oh, what's wrong with this patient, or like how can you help them? And like I wish I had done more anatomy in high school because it's like such an applicable thing to know. And they're like, oh, you know, like where is the gallbladder located, or like why exactly should you never put a patient in that position? And like it is really really cool to have this kind of knowledge. I just can't emphasize that enough because like it's really cool, a lot of the science Olympiad fields, but also because they don't get to be taught in high school. But of those things that aren't taught in high school, like the life science events, especially like public health and disease detectives, and specifically like anatomy and physiology, and learning these medical conditions is like a very applicable thing. So okay, moving on to disease detectives. The first part of disease detectives that everyone should get through as soon as they get thrown onto the event or whatever is to get the rote memorization out of the way. You want to be able to not only memorize term definitions, but to differentiate them, because a lot of them are really, really similar. The one that I can think of off the top of my head is the outbreak versus epidemic versus pandemic versus cluster. Right? An outbreak is more cases than expected in a certain area. A cluster is just a lot in a certain area. An epidemic is an outbreak over a wide geographical area, and a pandemic spans different continents. So it's a little bit different. You should definitely know how to differentiate these things. And here's one of the questions I had put on the MIT test, which is available online for free. And as you can see, I have a select all that apply question, and I've given 3 points to every select all that apply question, so nobody really knows how many to actually select. It could be one, it could be all of them. And in this case, it was 3 of the above. I forget which ones exactly. If I stared this for a while, I might be able to tell you. But it really tests, like, do you know how these definitions apply to this question? And disease detectives is a lot of application. I like to ask about vocabulary questions in this way, as in, like, which of these best fits this scenario. But a lot of people just go straight to word bank, and that is definitely also an option. And the best way to prepare for those, and how I prepared for this, is just a Quizlet. You can look at existing disease detectives Quizlets. Those terms have not changed in like 10, 12 years. And that is such a great place to start. And once you start being able to fill those terms in on yourself, by yourself, you get off to a great start. And you'll be able to start reading through the reports and the cases and really be competent with the kind of questions that are being asked. Oh, and also textbook glossaries, sorry. That means like, if you look up like the CDC textbook glossary, you kind of scroll through and make sure that you can identify all of those terms, and maybe make your own Quizlet that's a compilation of different textbook glossaries. That's a great way to start with these terms. I would say that for any life science event, any study event, like textbook glossaries. We'll actually go over that more in inquiry events when we talk about like Science Word and Picture This, because it comes in handy there too. Another thing that I've been like, disease detectives is a very practical event. It looks into epidemiology, which is public health, and the geographic and social distribution of diseases, who gets them and why they get them. But in that practicality, we also need to ask what actually happens in the field, because disease detectives at the national level is sponsored and written by the CDC, and they are really, really, really good at writing practical tests that put you in like scenarios and ask you what should you do. And in this case, I actually asked about a real life scenario that is actually from the World Health Organization ethical guidebook, and I asked, what is the correct thing to do here? So I'll actually open this up to y'all. According to the World Health Organization, is it ever ethical for doctors to provide individually identifiable data to health officials who request that data in order to monitor an outbreak? What do y'all think? let's do a show of hands. You can either do it, right? You can either do yes or no, and I won't judge you either way because I can definitely see reasons for both.
I would say
Yeah,
no. I would say no.
you would say no.
Yeah.
Okay, why?
I mean, unless doctor has some understanding with the patient and patient says yes, it
Yeah.
can be shared,
Yeah.
right? So that would be the first criteria, I believe.
So you're talking about informed consent.
Exactly.
So for show of hands, who thinks that this is ethical? And okay, sure. And who thinks that it's not? Okay, so it's about half and half, and that's actually the distribution of answers that I got on this exam. And the answer is actually, according to the World Health Organization, yes. Because what will happen is that individually identifiable data won't actually be used to link like a condition to a person. But what it will be doing is in a database, it will be eliminating duplicates and making sure that the population is well covered. And in that way, it really does help to have individualized identifiable data because that way we don't count the Joe Smith who lives at this address 3 times for his condition, which is extremely rare, and then we overestimate its occurrence in the population. And then in number 16, is it the norm to ask participants in public health surveillance, which is basically what epidemiology professionals do, is they talk to hospitals and doctors to see who's getting diseases and where they should be focusing their efforts. Is it the norm to ask participants in public health surveillance, so the people who are getting their data reported, for their informed consent in the surveillance? Give you all a couple of seconds to adjust that. Show of hands, who thinks yes? And who thinks no? Okay, so that's like a decent kind of half-half split again. And the answer is no, it is not. So, and this like this, no, exactly. But that's like really why I like to ask these questions, because these are the less obvious things and the less like common sensey things, but in a way they're still like very practical. What happens is that if you ask people for informed consent and surveillance, then people opt out. And when people opt out, then the statistics that you compile and the data that you compile is not representative of the population. So if you actually end up overestimating the amount of people who have like a very rare form of cancer, you might misdirect a lot of resources towards that instead of a more common form of cancer or something. So that's actually the World Health Organization recommended mode of action for that scenario. So what else was I going to say? So like informed consent in the surveillance, like you would think that like, oh, that's actually like really important, and honestly it is. And in most cases, especially for people with rare diseases, doctors will talk to their patients about submitting their data and stuff. But you guys are actually being surveilled right now, and your data for when you go into a hospital or a doctor's office and you submit a chief complaint, statistics are compiled, and that is how epidemiologists know what's going on, and that is how they know to post like on CNN News that hey, there's a romaine lettuce outbreak caused by an E. coli outbreak caused by romaine lettuce in like the Southwest United States, so be careful. And chances are not every single one of those people who had that condition, who just came into the hospital for a stomach ache, the chances are not all of them were told that their data was going to be used for that analysis. So anyway, I really like to go into that, and I think that that's a trend that disease detectives have been taking, disease detective tests have been taking recently as well. Another thing that I like to ask in my, and this is a little bit more unorthodox and a little bit more advanced, but I think that especially if you're in high school, you should be more familiar with experimental techniques used in disease detectives and in the lab setting. This is what is called an ELISA, or enzyme-linked immunosorbent assay. Very, very long name, but all that it means is that there are certain detectors on a plate. You put a biological sample on the plate, and then if there are certain molecules in that plate that react with your detection antibodies, there will be a color change. So it's basically like if I want to check if you have a very specific type of bacteria, and I know that the bacteria produces toxin A, then I'm going to put detectors for toxin A that will change color in the presence of toxin A. And that is actually how lab tests are performed in many cases to check if people have certain illnesses. And sometimes these color changes are too undetectable for our naked eye, but if you put them in like a spectrophotometer plate that can analyze the absorbance of a solution, it gets a little bit technical, but you're able to tell if someone has a particular strain of an illness or not. Does anyone have any questions? I doubt it. I doubt it. And the thing is, but sometimes people aren't afraid to mention this in the middle school report. So it wouldn't be essential to an answer. If I was to put in a middle school test, it would never be essential to an answer. But actually, if you look at any CDC report online, they always mention their detection mechanism or their testing mechanism. So just to vaguely have an idea that that's a testing mechanism, to know what it is, the textbook definition of it, even if you don't understand how the test works, that's a great starting point. That's a good question, really good question. Here's another way that people like to detect certain strains. Different strains of a single bacterium, so maybe a Staph aureus infection, could be a lot worse. And so one way to differentiate that is something called gel electrophoresis. And so you actually fragment the sample and you can see a little profile. And as you can see, the profiles that line up specifically, those are probably the same type of strain. But if you have some known strains and you know what they look like in the gel electrophoresis diagram, and then you go to some new country and you really want to test these people to make sure they don't have something you've never seen before, and you don't treat them for something that you have seen but it's not actually what they have, then you can run it and test the profile as well. So gel electrophoresis is also one of those common methods. I know this is mostly supposed to be for how should you study for this event, but I just want to mention and have y'all know that experimental techniques in disease detectives are definitely fair game. People ask about it, and honestly, I think it's really interesting too. So as we talked about this in anatomy, so I'm not going to put y'all through this, but pulling information from context and ugly long paragraphs is really, really important. So this was in my MIT test. This is straight from an actual CDC report. I would put this on both a high school or a middle school test. So I'm skimming this, I'm looking for numbers. As I'm looking for numbers, I look at 56% of interviews, 16 for whom professional language interpreters were interpreted. 28 to 92 days after the December 28th event, there was a questionnaire. Blah, 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 blah. 12 cases were confirmed. Sometimes people put the numbers written out, and competitors actually do miss those. And that's not any fault of the reporters, just sometimes people do that. So, and I'm also learning which numbers are important and which numbers to discard. I really don't care that 16 people had professional language interpreters. That comes into play somewhere else, but it's not going to be useful in my calculations. So as I go to the next slide, I've highlighted the most important part. So people who were interviewed versus people who actually had the condition. That's important. It's a special ratio. Anyone who's studying for disease detectives will know that. It's fine if you don't. And then I'm also seeing places where things could go wrong in a practical way. If professional language interpreters were used, they are professional, but things can still get lost in translation, especially with dialects. And this is actually these were actually people from a different country who had a celebration in California. So I can definitely see a loss of information during that translation. Contact information was unavailable from 14 potentially exposed persons. So what if those people would have changed our ratios and made us realize that this is a lot worse than we thought it was? And 2 people could not be reached. So are we going to follow up with those people? Who knows? Also, a questionnaire was administered by telephone 28 to 92 days after the event. So people are forgetting. These are all actual types of biases in disease detectives. Things that y'all, without ever having studied for disease detectives, could actually realize could be wrong, but they have a specific name, this is recall bias. Are people really going to remember exactly what they ate that night? Are they going to remember exactly how they felt or exactly how many hours they started feeling bad afterwards? Not really. So that's one source of error. And as you can see, there is a mention of ELISA. Actually, this is unintentional, but just in a random paragraph from a random case in the CDC weekly report, they mention the ELISA test. And as a middle schooler, you don't need to know the mechanism, but you shouldn't be completely confused when you see that. So where do we go for practice for disease detectives? Morbidity and mortality weekly report from the CDC is my favorite place to go. In fact, we're going to go there right now. That is actually where people who are trained publicists for the CDC issue the reports that they are working on right now. So before we go there, what do y'all think is on the forefront of the CDC's mind right now? What is something that y'all have heard of that has become particularly bad recently? I need to give you more hints. It affects young people indiscriminately. Yeah, go ahead. Yeah, y'all knew it instantly. 4 patients, yeah, and it's actually been a lot more. So that's actually where we're about to go right now. The most, and I guarantee you this year somebody is going to test you on this if you are in disease detectives. Characteristics of a multi-state outbreak of lung injury associated with e-cigarette use or vaping, United States. And this is just the first article that shows up if you Google MMWR. And so you see all of these researchers and PhDs came together to write this report. And this stuff is so cool. And so as I'm looking at this, we'll just look at the summary. What jumps out at you as y'all read this? You can tell me, but what pops out at you as things that I could ask a question about, or things that you should ask yourself as reading this article? Yeah, go ahead.
Oh, okay. 6-9% are males, which means that the majority of people getting this illness are male.
Yeah, so 7 out of 10 people who are having injury associated with e-cigarette use from male. A question we can ask ourselves is why? That's like a very common question. They might be like, propose 3 reasons why this could disproportionately affect males. And to be honest, I don't have the answers off the top of my head right now. But there are probably research articles that you can look up on that. And they honestly don't want to jump to too many conclusions in this article, especially because the outbreak is first being reported. But if you give this enough time and you revisit this topic in a couple of weeks or like a couple of months, you'll probably find more definitive explanations of those inequalities. What else do you notice? Yeah. Yeah. So 62% of patients were aged 18 to 34 years. So it's young people. It's not older people who are trying to use e-cigarettes as a way to quit smoking. It's young people who actually often use e-cigarettes as their first exposure to smoking. Yeah, all the percentages, right? THC was reported by 76.9%, but that doesn't exclude nicotine-containing product use. So obviously this is dangerous. They say the cause of the outbreak is unknown, but there are still things that you can be asked from this. And based on these numbers, these raw numbers, if I was to not give you these percentages, if I was to go and give you the actual numbers in the report, then you might be able to calculate some percentages from like attack rates, prevalences, incidences. These are all just terms in CDC that the CDC defines for epidemiology. So as I'm looking through this, I also see some more inequalities. I want you to ask y'all to read this whole thing. It's a huge chunk of text. But as of September 24, 2019, 46 state health departments and 1 territorial health department had reported 805 patients with cases of lung injury, blah, blah, blah. So that is 46 state health departments out of 50. So why did some states not report? What could be potential kind of biases that result in a report because of those 4 states that didn't report and the rest of the U.S. territories? As I'm looking through this, just so like I can boost my future knowledge, why are 4 states allowed to not report this? If it's like a CDC is a governmental entity, why can't they just find this out? And so like I would go and like look up that process as well. So like this is all like practical things that you should ask yourself, putting yourself in the shoes of an epidemiologist. And that is like the coolest thing to me. Median age is 23 years old. So yeah, we talked about the age gap and all of that. If we go to discussion, basically it's like blah 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 blah. Don't vape. Okay, so don't. Not that y'all needed to hear that. Anyway, yeah, you'll go there and every single like romaine lettuce outbreak or whatever that y'all see on CNN, any little outbreak, chances are it came from the CDC and chances are it's going to show up in the MMWR. Every single year I've done disease detectives, some current event shows up. In 2014, everyone was asking about Ebola every single time. So even though it wasn't really the topic for that year, and I think they've kind of moved away from topics per se, but even though it wasn't the topic that year, everyone asked about Ebola. So I just chose to be prepared. So that's one good way to prepare for disease detectives is be up to date with current events. Talk about it around the house. Talk about it in the classroom. It's such an interesting topic. Everybody thought that vaping was fine. Everybody in my high school was vaping in the bathroom between 6th and 7th period. And it's like, guys, what are you doing? Also, the CDC has an official online textbook where you will find every definition you will ever need that will take you from invitation to nationals. What happens at nationals is just you have to be better prepared to be a fast worker, and honestly, as you get towards state as well, be a fast worker and be a more practical thinker. If someone asks you what are 5 ways that this restaurant can improve their hygiene for the future, you need to remember all of the stuff you read in the case. You be like, well, they weren't washing their hands regularly. They weren't separating the preparation of the meat and the vegetables. Like, you need to be able to like think of all of that. That comes with practice, and that comes with reading MMWRs. And also, World Health Organization also has a textbook. They also have like random guidebooks and stuff like that ethical guidebook that I pulled that question about.
Can I add something?
Absolutely.
In regard to current events, those are also, I feel like current events are where your easiest points are in terms of, like, even in anatomy and physiology, if there has been a new technique to get biopsied or a new technique to detect cancer, it will be on the test. So making sure that you know your current events is really important for life science events because we're always evolving, right? And we're always evolving technology. And that's what they want to see on the test. They want to know that you're up to date and they want to know that you know your materials. So I agree, current events is really important.
Yeah, thanks. Does anyone have any questions before we move on? I think we're moving on to genetics just in the last 15-ish minutes. Okay. So genetics, I'm less passionate about. Disease detectives is my baby. But genetics is like the current structure of the rules is kind of not that great. Especially if like they kept it similar to last year's. I still have to look at this year's again, but last year was like a table, and correct me if I'm wrong for they changed this year, but it had like a bunch of random topics in genetics, and the wording of the rules says that the genetics testing is limited to these topics, is limited to topics including, right? So like that's really vague, and people could really ask you anything about genetics from like a middle school Punnett square level to like, a collegiate kind of recombinant technology level, and that's a little bit difficult to prepare for. But the way to prepare for that is to start at the foundations. Vocab is not so bad like disease detectives. Honestly, disease detectives vocab tends to be 3 points, honestly, in the most part. But foundations are essential, so good concepts. So the main things are like Punnett square computation and logic, which I think is part of many middle school curriculums, but not all. Not all. And that can be a great thing to help your students with because it's actually a little bit intuitive. It's like setting up a little multiplication table, and students are usually able to grasp that really, really well if you set it up like that table and stuff. And Punnett square computation can be extended to not just like one gene, but 2 genes, 3 genes with like a dihybrid cross. And so that all of that analysis is built on the 2x2 Punnett square. And so that is the first foundation that anyone should start at. Even high schoolers should brush up on that. Prokaryotes versus eukaryotes. That's a huge theme. We don't have the same genetic regulatory mechanisms. Prokaryotes are more efficient in some ways, but also more vulnerable to like genetic change in other ways. And emergent technology. This is really cool, and they focus more on it in Division C than B. But like CRISPR, gene editing, what people are coming up with these days is crazy. And so honestly, for genetics, you do want a relatively more up-to-date textbook. because CRISPR has evolved even between last week and this week. So yeah, let's get into it. Technology first. If you really want to test your mastery about something like CRISPR, you should be able to do like a Google image search and see a diagram of it and know what's going on. So from here, I can kind of surmise that there's a protein Cas9 that is joining to a gRNA. You would have to look up what that means. It's a guide RNA that basically tells it where to cut a piece of DNA. Then it cuts the DNA, and the DNA tries to repair itself, but it makes a mistake, and that mistake knocks out a gene. And that is how you can actually do like gene editing with CRISPR. And instead of knocking out that gene, you can use that cut to introduce a new gene as well. So like that's like CRISPR technology, but you should be able to understand different diagrams portraying the same thing. Because and that was actually my way of studying for a lot of this stuff. I actually never got to compete in genetics. It extended to a lot of my other events, but I did get to write a test for this for Princeton, and I definitely went into the CRISPR-Cas9 system. But I did try to give a lot of background information because I realized that not all schools teach it the same, etc. So yeah, you have to also try to understand mechanisms, not just functions. It's really easy to go, just like in disease detectives, it was always a good practice to ask yourself why as you were reading. Why are there these inequalities in who is vaping and who is getting injured from vaping? And why are there these inequalities with who is reporting vaping injuries? Like which states? Now, here, you might read in a textbook that a repressor is bound to the DNA at the operator. If you look at 8b, I give you that information. And then I ask, how does this repressor prevent transcripts from being produced? Right? So then you would know that like it physically prevents a different protein, a specific protein from binding, and you would give me that, and I would give you a point there. But actually, like in most textbooks, you'll just get like the first thing, but you should always ask yourself kind of why. I'm not going to go into that too much. That I feel like that's kind of given knowledge, and I'm not going to keep emphasizing that for no reason. So where do we go to study genetics? A great place to start for any life science event, except for stuff like disease detectives that is so specialized that there's already a really good resource out for that. Campbell Biology, any recent 8+ edition, and if you were in the study resources talk, we talked about 8th edition Campbell textbooks going for like probably $5 used or even less online, because at this point they're at like 11th or 12th edition.
Yeah.
So you can use a teacher's help, like go to tutorials of actual biology teachers at your school and stuff to help understand every diagram and every term. And it's such a good exercise to try to work through a textbook that is a little bit above your understanding level, and then kind of bring it back down to your understanding level and paraphrase things in ways you can understand them. And that actually helps with retention so much. Advanced students, once you have what is in Campbell's under your belt, though I have to say Campbell's is going to cover such a huge portion of what you need to know for genetics. You can go look at Hartwell Genetics, which talks a lot about like recombination, new technology, really, really cool stuff. Does anyone have any questions about these resources? I've kind of been flying through. So, okay, and we'll end with just talking about taxonomy in general. We touched on taxonomy a little bit during the binder building section. That's really the smartest thing you can do for taxonomy is build a good binder. And we talked about that in the study event resources part. But it requires very intimate knowledge of your binder. So you must assemble your binder or your notes. You must know how to flip to something like, and you should go to a competition, and if you were like, you know what, that was really, really inefficient, talk to your partner and figure out a new organizational scheme. Taxonomy also requires reliable spot identification of specimens. And one specimen could look like a lot of different things. And that's what I learned the hard way in Rocks and Minerals. And Yen was also talking about that earlier when she was saying how she used to actually go places and go to like museums or festivals or fairs or science events to get to know all the different ways like, you know, a rock could look or a rock could be produced. And so, yeah. You should also have like an ability to understand how taxonomy and ecology fit together, especially for those events like ornithology, how like climate change is affecting the distribution of certain things, how tectonic shifts affect like the distribution of rocks and rock types, So everything ties in together. It's easy to get so overwhelmed with the memorization component of taxonomy events that you don't realize how it connects to other fields of science. But that is probably one of the most important themes, to be getting those extra kind of difficult questions right in taxonomy. Does anyone have any questions about? Does everyone know what taxonomy is in the first place? It's just like that rote memorization, kind of classification of ornithology, rocks and minerals, water quality, kind of ecosystems, like you name it. So, I think that is about all that we have for study events. So y'all have about like 10-ish minutes to just chill and get some water, get some food. We still have food and coffee. It's cold now, but yeah. Does anyone have any last-minute questions?
I'm also the lab events coordinator, so if you have any questions about lab events, I can answer those too.
Yeah. All right. Thank you all so much.
Thank you.